Genetic engineering is changing the world around us. It is a powerful tool that allows scientists to alter the genetic makeup of living organisms. This technology has the potential to revolutionize many fields, from medicine to agriculture. Now, it is being used to help conserve endangered species, including the golden tiger. Golden tigers are incredibly rare. They get their unique coloration from a recessive gene. This gene disrupts the production of black and brown pigments in their fur. This results in a beautiful golden coat with pale stripes. Scientists are exploring the possibility of using genetic engineering to increase the number of golden tigers. Gene editing technologies like CRISPR-Cas9 have made it possible to make precise changes to an organism's DNA. With CRISPR-Cas9, scientists can target and modify specific genes. This technology could be used to introduce or enhance the gene responsible for the golden tiger's coloration. This would increase the chances of breeding more golden tigers in captivity. However, genetic engineering is not without its challenges. There are concerns about unintended consequences and the potential for unforeseen problems. It's crucial to proceed with caution and carefully consider the ethical implications of such interventions. The welfare of the animals and the preservation of natural biodiversity must always be paramount. Breeding rare species like the golden tiger is a complex undertaking. One of the biggest challenges is the issue of genetic bottlenecks. Small populations like those of endangered species often have low genetic diversity. This is because they have a limited number of individuals contributing to the gene pool. Low genetic diversity can lead to a number of problems. It can make the population more susceptible to diseases. It can also lead to reduced reproductive fitness. This means that animals may have fewer offspring, or their offspring may be less likely to survive. Inbreeding, which is more common in small populations, can exacerbate these problems. Another challenge is the technical difficulty of genetic engineering itself. While technologies like CRISPR-Cas9 are powerful tools, they are not without their limitations. There is always a risk of off-target edits. This is when the gene editing tool accidentally alters the wrong part of the DNA. Unintended mutations can also occur. These mutations can have unpredictable effects on the organism. Even if the genetic engineering is successful, controlling gene expression can be difficult. Gene expression is the process by which the information encoded in a gene is used to create a functional product, such as a protein. The environment can influence gene expression. This means that an animal with a genetically engineered trait may not always express that trait in the way that scientists expect. The use of genetic engineering in wildlife raises a number of ethical concerns. One of the most pressing concerns is animal welfare. Critics argue that it is wrong to manipulate an animal's genetic makeup for human desires, even if it is done with the intention of conservation. There are concerns about the potential for unintended health consequences and suffering for the animals involved. Another concern is the idea of human interference in nature. Some people believe that we should not interfere with the natural world in this way. They argue that genetic engineering represents a step too far. They worry about the potential for unforeseen ecological consequences. They also worry about blurring the lines between natural selection and human-directed evolution. The potential for commercialization of genetically engineered animals is another concern. There are worries that this technology could be used to breed rare animals for profit. This could undermine conservation efforts. It could also lead to the exploitation of animals. The focus should always be on the well-being of the animals and their ecosystems, not on economic gain. The ability to manipulate the genetic code of living creatures raises profound philosophical questions. Some view genetic engineering as a form of playing God. They argue that we are exceeding the boundaries of ethical human intervention. They believe that we should not tamper with the fundamental building blocks of life. Despite the challenges and ethical concerns, genetic engineering holds great promise for conservation efforts. In the future, it could be used to help save endangered species from extinction. Scientists are exploring a number of potential applications. One promising application is using genetic engineering to enhance disease resistance. By introducing genes that confer resistance to certain diseases, scientists could help make endangered populations more resilient. This would be particularly useful for species that are threatened by infectious diseases. Another potential application is improving reproductive fitness. Genetic engineering could be used to address genetic bottlenecks and inbreeding in small populations. This could involve editing genes to increase genetic diversity or to correct genetic defects that reduce fertility. In some cases, genetic engineering could even be used to resurrect extinct species. While this sounds like science fiction, it is becoming increasingly possible thanks to advances in gene editing technologies and our understanding of ancient DNA. However, it is important to proceed with caution. The long-term consequences of genetic engineering are difficult to predict. It is crucial that any interventions are carefully considered and carried out with a focus on the long-term health of the species and their